Okay. Hello, my name is Crystal Santos,
And I'm Helena Maya.
and the topic that we ended up choosing is DNA printing.
Okay, what is DNA printing? DNA printing is a laboratory technique that is used to determine the identity of a person based on their nucleotide sequence, which is unique to each individual. It's a process of matching two DNA fingerprints with one another, such as DNA samples from a known person and one from an unknown person. It's a method used to help narrow down lists of suspects in a crime. Some examples are solving crimes, establishing paternity, and identifying disaster victims.
So, how does DNA printing work? DNA printing works as you gather samples, as in blood, saliva, skin tissue, anything that's involving in a human body. As you gather those samples, you can collect the DNA from the cells, which also includes cell lysis that separates the DNA from debris. Amplification is provided to make sure enough DNA is added to make an analysis, which is used by PCR. The sample is ran through a capillary that contains a gel, which allows smaller sequences to move faster through pores. And once those processes are done correctly, you compare and interpret the results. You either study the differences or if there are any matches to what you are studying, which I can also include electropherograms in whichever sample you have created.
What does DNA printing create? It creates a genetic code that eventually becomes a computer code. This normally occurs when chemical bases like adenine, thymine, cytosine, and guanine present in a chemical mix. These genetic codes translate into computers through electrophoresis technology into the representative letters. These letters are AT, TA, CG, and GC. DNA printing is also known as a method in synthetic biology that is used to create artificial genes.
So, DNA printing is used for identifying unknown bodies, discovering diseases, and blood relatives. DNA printing is used mostly like during like while you're studying bodies or in like murder scenes, crime scenes, and figuring out any missing pieces from a crime scene. And it can also give the professionals who are studying the DNA printing samples a higher chance to know who did what and who was the victim. DNA printing is also used for finding out any sort of diseases that you might have in your system that you don't know about. And it can also give you a higher chance to know what the like disease is and how to cure it. Some challenges for DNA printing can be collecting DNA since it's not always easy to know what the DNA is or what to collect at the time, and which can also involve misleading results, cross contaminations from different DNA samples, and the samples can also be ruined by light, moist, or heat. Some other challenges involving DNA printing, as in the control, it can be in the environment. The control is to maintain the sample stored and decrease the risk of any degradation. Another challenge is the technical issues with equipment being used, and PCR can introduce any sort of error which complicates the analysis of the DNA printing samples.
Some improvements that have been made to DNA printing is increasing the speed of DNA printing, natural enzymes for longer and more accurate results, and laboratories now have long-term DNA printers and automated systems which reduce any errors.
So some limitations to DNA printing. Some programs limit the amount of handling human DNA printings. Some samples are mixed together, which causes the process to have complications and having the samples to be redone. And mishandling samples can also give false information due to professionals not being careful with the samples or just not caring enough for the samples, which is what causes false information. And since those things are done and it is possible, it also like doesn't always prove who is at fault of who did what.
Some interesting facts to DNA printing are identifying non-code regions of DNA. Some researchers are finding fast methods every day to solve DNA analysis. And if you didn't know, the human genome is made up of about 3 billion nucleotides, which are 99.9% identical from one person to the next person. Um, and DNA testing is used to authenticate expensive foods like caviar and fine wine.
So here are our work sites, and that is our project.